Thank you very much, the organizers, for giving me the chance to present this abstract. So, this is an original article published in Cancer in year 2020, which showed basically the effect of MEK or ERK inhibition, which effectively abrogates the emergence of acquired osimertinib resistance. In the last few minutes, we were discussing about various pathways for osimertinib resistance. So it is definitely a very pertinent issue with us. And this original article is discussing the MEK or ERK inhibition, which can effectively abrogate the emergence of acquired osimertinib resistance in the treatment of EGFR receptor mutant lung cancers. The principal investigator was Diaju Gu, and Dr. Suresh Ramalingam was also part of the investigatory group. So the background of this trial was that osimertinib irreversibly inhibits EGFR exon 19 and 21 and also the resistant P790M mutations. Based on this fantastic response, it has been approved for use in the first line, second line EGFR mutated NSCLC. However, we all know that the patients eventually relapse from osimertinib treatment because of the development of acquired resistance. So before starting the abstract presentation, I would just like to revise the various signal transduction pathways which are there. So this is the signal transduction pathway in an EGFR sensitive cell, whereby if we kind of block the EGFR receptor, the downstream signaling cascade is effectively blocked, leading to a very effective apoptosis of the cell. But in a resistant cell, there are many resistance pathways which develop. One of them is MEK, one of them is HER2/neu, another one is RAF mutations. So all these kind of appear after prolonged use of osimertinib. So they can lead to either a primary resistance to osimertinib or more commonly secondary mutation to osimertinib. And hence the logic of adding a MEK inhibitor, trametinib, or an ERK inhibitor to kind of complete the vertical blockade of the same pathway. So the trial methodology in this trial was basically, it was a lab cell line based study and a mouse xenograft based study. So the drugs used in this trial was osimertinib, trametinib, which is the MEK inhibitor, and ulixertinib, which is the ERK1/2 inhibitor. Now, coming to the results of this trial, we can see this. This is a cell line blot study, whereby we can see that osimertinib exerts transient inhibitory effect on the MEK-ERK signaling in comparison with sustained suppression on AKT. And with passing of time, the inhibition of the downstream ERK pathway is subsequently decreased. So that is the logic of actually. So that is the logic of adding a MEK-ERK inhibitor for a sustained inhibition of the entire signal transduction pathway. So in this, we can see that with single agent osimertinib, there is definitely an upregulation of ERK, which can be blocked by giving a ERK1/2 or a MEK inhibitor. So this was another important result which was obtained from this study. So this is again a cell line plot, a bar plot, whereby we can see the extent of apoptosis in the various cell lines. I would just like to focus on the left-hand side of the curve. In this, basically, we can see that with osimertinib alone, the extent of apoptosis is only in the range of 20% in this particular cell line. If we add osimertinib and trametinib, the trametinib is the MEK inhibitor, definitely the chances of apoptosis increases and it approaches approximately 45%. And if we are able to add on ulixertinib, which is a ERK inhibitor to osimertinib, the chances of apoptosis increases beyond 50%. So there is definitely an improvement in the rate of apoptosis if we are combining osimertinib with MEK-ERK inhibitor. So this was another important result which was obtained from this study line, which showed that there are intrinsically osimertinib resistant cells which are present within sensitive EGFR mutant cell population. We all know that every tumor is heterogeneous. They carry various cell clones. Some of them may be sensitive, but few are resistant at upfront presentation. So here also in this graph, we can see that there are a clone of population which is resistant to osimertinib. They just don't respond to osimertinib. So hence the rationale for adding means osimertinib combined with a MEK or ERK is highly active in killing this intrinsic osimertinib resistant clones through the induction of apoptosis. So this is a mouse xenograft model whereby they have the investigators have calculated the amount of the tumor growth. So here in this graph, we are seeing that this green graph is represented by a single agent trametinib alone. The blue graph is representing osimertinib alone, and the red graph is representing the combination of osimertinib and trametinib. And here we can see that the tumor size is minimum in the combination treatment arm, and hence the combination of osimertinib with MEK inhibition concurrently or intermittently prevents or delays the emergence of acquired resistance to osimertinib in vitro and in vivo also. So this is how we can see is the improvement in the cell kill with the combination of osimertinib and MEK inhibitor. So after these important results, basically we can logically say that there are two strategies to adding trametinib to osimertinib. Either we can do a salvage combination approach of osimertinib plus MEK inhibitor after a patient develops resistance to osimertinib. So that is one approach. The second approach is we start osimertinib and a MEK inhibitor or ERK inhibitor right from day one. It's a combination approach upfront to delay or prevent the resistance of osimertinib. Which one of this is more kind of attractive? The investigators concluded that this approach is more attractive, whereby we can use combination approach right from day one of osimertinib and the MEK ERK inhibitors to prevent or delay osimertinib resistance. So the conclusion from this trial was the findings from both the in vitro cell line models and the in vivo mice models in this study have demonstrated that targeting the MEK ERK signaling by trametinib or ulicertinib effectively prevents or delays the emergence of osimertinib resistance. And the authors have also tried various intermittent dosing schedules of osimertinib and trametinib, which effectively delays the resistance without causing undue toxicity to the mice, to the xenograft model. So food for further clinical studies is that this study has given us a strategy that we can definitely improve upon the results of osimertinib by adding onto osimertinib MEK inhibitor or a ERK inhibitor. And we all know that a similar strategy on vertical blockade of the signal transduction pathway has already been successful in BRAF MEK inhibition in lung cancer. Thank you very much. Thank you. Open to questions.